hope you are fine. You are fine. Is my voice clear? Today I want to talk about the continuance of a previous lecture, class Ascomycet fungi. Compatibility in Ascomycet fungi. Compatibility actually means fusing capability of sexual nature of the mycelia. According to this, Ascomycet fall into two distinct groups. Homothallic species. Several individuals are able to form asci themselves without the aid of other mycelium. They are therefore self-compatible or self-fertile. The mycelium in homothallic species considered bisexual, but heterothallic species where two compatible individuals mated before asci form. Therefore, the mycelium in this species is unisexual. In homothallic species, mycelium considered as bisexual, but in heterothallic species, the mycelium considered unisexual. Alternatively, it may also say that sexually it is self-sterile, and for sexual reproduction, it requires the aid of another compatible mycelium of a different mating type. The two compatible mycelia may be named plus and minus strain. Two compatible mycelium that this mycelium are from different mating type. These two compatible mycelia may be named plus and minus strain mycelia. There can therefore be a fusion only between a plus ascogonium and minus or antridium. Ascogonium is female gametangia and antridium is male gametangium in Ascomycet fungi. Or a ascogonium and antridium. This phenomenon called heterothalys. In homothallic species, species are self-fertile. The mycelium in homothallic species considered bisexual, but in heterothallic species, two compatible individuals mated before oosphy formed. The mycelium in this species is unisexual. For sexual reproduction, these species require another compatible mycelium from a different mating type. Two compatible mycelia may be named plus and minus strain mycelia. There can therefore be a fusion only between a Ascogonium and Antridium. That Ascogonium is a female gametangia and Antridium is male gametangia. In heterothallic species, two compatible mating type or MAT1 and MAT2 require for sexual reproduction. MAT1 and MAT2 fusion and produce a eukaryotic mycelium. Nuclei comes from MAT1 and MAT2. One eukaryotic cell produce mother cell ascus. After karyogamy, the ascus form and these os contain 8 sexual spore or ascospores. Ascus development. Ascus develops after fertilization by direct or indirect method. Direct development of ascus. In lower ascomycete, is my voice clear? Okay, thanks. In lower ascomycete, plasmogamy followed immediately by karyogamy. Resulting in the formation or a diploid nucleus. This is the eukaryotic cell. After plasmogamy, this eukaryotic cell contain two nucleus from MAT1 and MAT2 produce a eukaryotic cell. Will the slide change for you? The cell containing this diploid nucleus developed directly into an oscus. This nucleus divided first mitotically and then by ordinary division, resulting into 8 haploid nuclei with change into same number of ascospores. Yes, Miss Adnan. Okay, I refresh the voice. Is the voice got better? Okay. Ascus development after the fertilization by direct or indirect method. Direct development of ascus. After plus follow after plasmogamy, resulting in the formation or a diploid nucleus. Plasmogamy and immediately karyogamy the diploid nucleus formed after karyogamy. The nucleus divided first mitotically and then by ordinary division, resulting into 8 haploid nuclei. That these haploid nuclei change into same number of sexual spore or ascospore. Do you have my voice? Okay. Indirect development of ascus in higher ascomycetes. The ascus development is indirect. The two gametangial male and female or antridium and ascogonium contact with each other. The male nuclei from the antridium and through the trichogen into the ascogonium. This is male gametangia or ascogonium, and this is antridium or male gametangium. A special hypha formed between ascogonium and antridium. The male nuclei from the antridium pass through the trichogen into the ascogonium. Female nuclei and male nuclei in ascogonium and get themselves paired with the female nuclei present therein. However, there is no fusion of male and female nuclei at this stage. This paired nuclei called dicaryons. In dicaryon or dikaryotic cells, two different nuclei pair together. On this stage, when many dicaryons are present in the ascogonium, the wall of ascogonium gives rise to many papilla-like outgrowths, which develop into short ascogenous hypha. The paired nuclei or dicaryons migrate into this ascogenous hypha. Young ascogenous hypha are accepted, but later on they become multicellular and profusely branched. Only rarely they are unbranched. Ascogenous hypha remain intertwined with one another. A majority of their cells are binucleate. Of these two nuclei of the cells of ascogenous hypha, one of is of antridial origin and the other is of ascogonium origin. The asci develop the types of ascogenous hypha. The terminal cell of each ascogenous hypha contains two nuclei. It preserves and forms a crozier or hook. This is dicaryon or dikaryotic cell. This nuclei divided and then septate formation. This cell elongate and form mother ascus cell. Karyogamy. And immediately meiosis and then mitosis and finally 8 sexual spore or ascospore form in asci. Both the nuclei of crozier divide to form four nuclei. Wall forms in such a way that a uninucleate tip cell, binucleate cell, and a uninucleate basal cell. Uninucleate basal cell. Binucleate penultimate cell become the oscus mother cell. It enlarges and develops into an oscus. The two nuclei of this oscus mother cell fuse and form a diploid nucleus. This diploid nucleus divides first meiotically and then mitotically, forming four haploid nuclei. One more mitotic division results in the formation of 8 haploid nuclei. All these nuclei surrounded by some cytoplasmic content and change into 8 ascospore. Asci and ascospores. An ascus is the sexual spore-bearing cell produced in ascomycete fungi. Asci are different in shape, may be elongated, oval, globose, club shape, cylindrical, or even tubular. The arrangement of ascospores in an ascus is also variable. They may arrange in uniseriate, biseriate, fasciculate, or irregular manner. On average, asci normally contain 8 ascospores, produced by a meiotic cell division followed in most species by a mitotic cell division. However, asci in some genera or species can number 1, 2, 4, or multiple of 4. Ascospores are non-motile, usually single cell, but not infrequently may be septate and in some cases septate in multiple plants. Mitotic division within the developing spores populate each resulting cell in septate ascospores with nuclei. It may cases the asci form in a regular layer, the hymenium. In many cases, the asci formed in a regular layer that called hymenium or hymenium in a fruiting body that is visible to the naked eye, here called an ascocarp or ascoma. For example, in Cleistocesium. In other cases, each fungi, such as single-celled yeast, no structures found. Asci normally release their spores by uh, bursting at the tip, but they may also digest themselves, passively releasing the ascospore either in a liquid or as a dry powder. The form of the ascus, the capsule that contains the sexual spore, is important for classification of Ascomycota. Three forms of asci: cylindrical, clavate, and globose or spherical. Ascospore shapes. The ascospores usually are unicellular and different shapes: triangular, globose, irregular with lateral ball, elliptical, almond, almond shape, narrow fusiform, broad fusiform, and lemoniform. Ascospores, ascocarps. In a majority of the ascomycete, except yeast and some related fungi of endomycetal, the asci remain enclosed by many sterile hypha. The Ascogonium, Ascogenus hypha, asci, ascospores, paraphyes, and the enveloping sheet or predium in ascomycete remain grouped together in the form of a fruiting body or fructification called ascocarp. Main type of ascocarps. Four types of ascocarp met with in Ascomycotina. Cleistocesium, Cleistocesium. Cleistocesium is globose fruiting body with no special opening to the outside. The asci arranged irregularly within them. When matured, the Elistothecia browsed open to release their asci and ascospores, as in Erysipelas or Eurotiales. Eristothecium is closed without opening pore. Apothecium, a cup-shaped fruiting body, as in Helotiales and Pezizales. Cup-shaped fruiting body. Apothecium is a cup-shaped fruiting body. Tritecium, a flask-shaped fruiting body, opening with an ostiole or pore, and its asci are exist in an irregular layer that called hymenium. Tritecium is flat. On type of this, and the ascospore release from this pore. Pseudothecium, a tritecium with bitonicate asci or two-layer asci, as in loculoascomycete. Some call pseudothecium as ascosteroma. Pretestial hypha, ostiole, neck, inner wall layer, outer wall layer, middle wall layer, rosette of asci, and the strial hypha or paraphyseal hypha that exist between the asci. They help to release the asci from the ascocarp in these fungi. Economic importance of ascomycet. Some ascomycets are of tremendous importance to man. Some examples of their determinantal are nature maintained below. Species of Aspergillus and Penicillium cause great damage to food and to some common goods such as leather. Leather can attack by Aspergillus and Penicillium species. Cadmium is a genus of Ascomycota. Destroy the fabrics containing cellulose, such as paper. They attack many crop plants as well as timber and ornamental plants, causing disease such as powdery mildew, apple scab, foot rot of cereal, and chestnut blight. They cause many diseases of domestic animals as well as man. Aspergillosis is caused by Aspergillus fumigatus, Aspergillus flavus, and Aspergillus niger. Claviceps purpura contain the sclerotia. There are sclerotia in Claviceps purpura. With many such alkaloids that it consumed are deadly to animals and human beings. On the contrary, it's used as a medicinal drug. Some of the beneficial aspects of ascomycet maintained below. Many yeast species, well known for their fermenting activities in brewing and baking industrials. Antibiotics such as penicillin are prepared from Penicillium notatum and Penicillium chrysogenum. Ascomyces members prepare many industrial products such as citric acid, oxalic acid, gluconic acid, vitamins, and glycerol. Some of the morels and troughs are edible. Some ascomycet used as a source of protein for human consumption. Classification of Ascomycotina. Do you agree to have a short break? 10-minute break. Classification of Ascomycotina. The main criteria of the classification of Ascomycotina have been type of structure of ascus, type of ascocarp, ascocarp centrum or ascocarp opening pore, and ascogeno and conidial stage of the organisms. Owing to the absence of fossil evidence and lack of our present knowledge regarding the developmental stage of this large group, the statement made by Alexopoulos and Mim in 1979 about the taxonomy of Ascomycotina appears quite convincing. According to Alexopoulos and Mim, the class Ascomycet is subdivided into the following subclasses: Hemiascomycetidae, Pelectomycetidae, Hymenoascomycetidae, Lobule, Benio. Do you have my voice? Okay, thanks. Lobule, Benio, Mycetidae, Locule, Ascomycetidae. Subclass Hemiascomycetidae. The Hemiascomycetidae include the simple ascomycet that some mycologists consider primitive and other degenerate. They have very simple structure. Sexual reproduction takes place by the conjugation of two cells, gametangia, which in some species are like Eremascus and in other unlike. The fusion cell be directly transformed into an ascus or grows up to produce an ascus. Characteristics of the do you have my voice? Okay. A complete absence of ascocarp, direct development of asci, Without the intervention of Ascogenus hypha, asci become directly on the mycelium or formed from a specialized Ascogenus cell. Subclasses Electromycetidae. General characteristic. There are important for final exam. The mycelium is well developed, branched, and septate. Many members show degeneration of sex organs, especially of male gametangium. The mycelium forms well-defined fruiting bodies or ascocarp. Asci develop from Ascogenus hypha, which are typically eukaryotic. The asci are typically 8-spore. In a majority of the members, the ascocarp is of Cleistocesium type, but some also have Apothecium or Ascosteroma. Cleistocesium not usual. Paraphyses are absent in the fruiting body. Subclass Hymenopteromycetidae. Based on the gross morphology of the ascocarp, the Hymenopteromycetidae comprises numerous species, which are under two groups, namely Pyrenomycete and Discomycete. Pyrenomycete characteristic. The characteristics of each subclasses are important for final exam. It occurs on wide variety of habitats such as dying, decaying stems and leaves, and soil, and some occur as plant pathogen. The asci are unitunicate or one layer and club-shaped or cylindrical and remain arranged in a hymeneal layer. Usually the asci are persistent, but they may also be evanescent. Majority of them develop ascocarp of a Pretium. However, some members contain Cleistocetial type of fruiting body. The pretaecium usually containing an apical opening called ostiole. The pretaecial ascocarp usually containing an apical opening that called ostiole. The pretaecium either occur singly or remain clustered together on or within a stroma. The ascocarp remains surrounded by a definite peripheral wall called peridium. Sterile filaments called paraphyses may also be present among the asci. Such filaments are absent in the plectomycete. Paraphyses grow into the central cavity, become enlarged, and provide a space where the asci and ascospore were developed. Ascospore often forcibly ejected from the ascus and pretaecium when mature. Species in this series may produce pretacia directly on their substrate or in a stroma. There are discomycete characteristic. I correct this slide. Members are either terrestrial, coprophilous, saprophyte, or parasite. Most members lack conidiophores and conidia. All non-lichen-forming discomycete have unitunicate asci. Color of the apothecia may be yellow, red, brown, or even black. Some are even hyaline or without color. The fruiting body of some members are underground or hypogean, and in other members are above the ground or epigean. The hypogean species grow underground and epigean grows above the ground. Members show progressive reduction in their sexuality. The antheridia and ascogonia are both functional in some members, while in the other the antheridia are non-functional. In other the antheridia are totally absent. The ascospores in all discomycete. Yes, all of the general characteristics of subclasses in the Ascomycota is important for final exam. The ascospores in all discomycete except tuberels ejected forcibly through different types of apical opening. Next week is an holiday. Uh, 2 weeks later, I will tell you the important parts. Do you have my voice? Okay, I told. 2 weeks later, I will tell you the important parts of the PowerPoint. You only have to read this question for the final exam. Okay? The lesson is over today. Do you have any question? If there is no question, you can leave the class.